saying here. So Leo is joining us from Finland, and he's presenting online on orchestrating microbiome multiomics with bioconductor. Thanks, Leo.
Okay, thank you very much. Let me share my screen. Wait.
Should I help now?
Almost there.
Samuel, you're here present?
Yeah.
Okay. Sorry for the late start, everyone.
Do you see my screen?
No, not yet.
Okay. Sorry, I thought I was the last one.
Levi, can you do? Have you better access to share Leo's slides there?
I can do it if you'd like. If you send me somehow a link, I can do that, but you probably should be able to share. Sorry, this is your punishment for being the first one who appeared ready, Leo.
Yeah, now I think I manage, maybe.
Sorry about this, Leo.
Do you know?
There you go. You're
okay,
good.
You see it now? Okay.
Yes.
Great. Do you see the full screen or only the slides?
Only the slides.
Excellent. Okay. All right. Thank you very much. And thanks for this opportunity to present our recent work on microbiome data science and microbiome data integration using the state-of-the-art data containers in bioconductor. And this is, of course, joint work with several people. I come back to that. But let's start by thinking for a moment about the needs to organize multi-assay microbiome datasets. So in a typical microbiome profiling study, usually we traditionally deal with taxonomic profiling. So in that case, the abundance matrix would contain the abundances of different taxonomic units across samples. But in addition to the abundance tables, we usually have rich site information about the features and the samples. And we, of course, need to interlink these with the abundances. But not only that, we have usually multiple assays that may come from, let's say, different statistical transformations of the data, different measurement technologies like 16S and metagenomics, and we can have different pre-processing pipelines. There are many different sources of the data that we want to link together. And these data sets are often hierarchical, meaning that we can have phylogenies in the feature space. For the taxonomic units, we have the phylogenetic trees, but we also regularly can have hierarchies in the sample space. So we can have, for instance, nested study designs, or we could have host phylogenies. So the tree summarized experiment data container in bioconductor is well suited for integrating all these different layers of a typical taxonomic profiling study together. This tree summarized experiment class is quite recent. It was published in 2021 by Ruichu Huang and others. And it extends the single cell experiment class. And basically, we have added here the tree information on the features and samples. And this allows us to consider the various hierarchies in microbiome data. So there is a clear need to organize hierarchical multi-assay microbiome datasets, and this container will help with that. In a usual microbiome data science workflow, we start by importing the raw data files into this particular data object, and then we have an ecosystem of tools that understand this data object for reproducible analysis of the data. This is, of course, the standard workflow for bioconductor analysis applied in different applications, but we have now started to build the microbiome ecosystem around the 3 summarized experiment container. And the advantage of this container compared to some former studies, similar studies, is that it provides enhanced ways of integrating the different types of the data. All right. Whereas much of the traditional developments in microbiome research in bioconductor have focused on the analysis of taxonomic profiling studies, there is now more than 100 packages listed on my colleague's website, Sudarshan Shetty's website, for this purpose. And there is now steadily increasing need to integrate the taxonomic profiling datasets with other omics datasets, including, for instance, transcriptomics, metabolomics, other functional measurements, or host omics, which then, of course, have different other complementary classes in bioconductor. So we use usually the 3 summarized experiment object for the individual omics, or we can use other classes from the bioconductor for the individual omics. And then we use the multi-assay experiment, which is an established class in bioconductor, for integrating these different types of omics. And of course, this idea is by no means specific to microbiome analysis. It is applied in different applications in bioconductor, but we have now taken advantage of this in the particular context of microbiome research. And there are different varieties of the data integration depending on whether the samples match, whether the features match, and how well they are interlinked. So we can utilize different aspects of these containers depending on the case. All right. This can have various benefits for microbiome data science compared to the more traditional ways of doing this in bioconductor. So it considerably extends our capacity to integrate data from multiple sources and multiple omics into microbiome studies. These containers and methods around them are also optimized in terms of speed and memory, allowing us to scale up the studies to larger sample sizes and cohorts more efficiently. And this is also integrated with other applications and frameworks. For instance, the single cell community and transcriptomics community are often using quite closely related data containers in bioconductor, meaning that we can cross-utilize these tools across the different application areas very efficiently. And we are already taking a lot of that into account in order to not reinvent the wheel. This, of course, makes all the development work more efficient. In the current package and data ecosystem that we have, we, of course, provide all the tools, different tools for basic microbiome data wrangling and all the basic analysis. But now increasingly also for the cross or joint analysis of the different data sources like taxonomic and metabolomic measurements, for instance. Unfortunately, time is too limited to give many details, but I want to give a short example soon. In addition to the methodology, we of course have the importers for the common file formats for microbiome research, which makes this system interoperable with many other tools that you can utilize for microbiome data science. And we also link to a number of open data resources for microbiome research. So, for instance, the independent projects on curated metagenomic data and Magnify API, and then some bioconductor packages and experiment hub. These provide collectively hundreds or even thousands of open source microbiome datasets that can be directly accessed by using the tools that support this ecosystem. So we can use this for demonstration purposes and research purposes quite efficiently. Just as a minimal example, this short snippet of code shows how you can download some open microbiome dataset from Experiment Hub, and then we agglomerate the taxonomic units to the family level and combine the rare taxa into a single group, and we transform the counts with CLR transformation, cross-associate or cross-correlate the selected assays from the microbiota and metabolite measurements, and ultimately draw a heat map. So this is just one single example, and about the cross-correlation, usual exploratory stuff for microbiome analysis, and there are also other multi-omics tools available now in the ecosystem. This just demonstrates a little bit how you can utilize the current tools. There are more tools available and documented in the online kitbook that we have been building. This is still a beta version, but you can find many workflows and examples from this Orchestrating Microbiome Analysis book. And it complements the other available bioconductor kit books that are out there and provides largely overlapping methods and analysis strategies. You can have a look at this online book for more details. And finally, of course, I would like to thank our open community. We have currently more than 20 contributors from around 10 countries. And of course, I would like to thank for the people and developers who are supporting data sources, building these containers, and providing other packages that support the ecosystem. You can find us from the Bioconductor Slack Miaverse channel, and more information about these resources is at the project website that you see here. Thank you very much.
Thank you so much. Have we any questions? I'll just take one question in the interest of time. Okay. Leo, what we may do is have questions afterwards. In the interest of just keeping things, the remote and the in-person sessions going, I think we're going to have a second remote talk and then do both of the in-person talks. So can I ask Simone Barry to talk about a novel statistical method for single isoform proteogenomics inference?
Let's see if I can share.
Wonderful, thank you.
Screen. Hey, can you see the slides?
Yes.
Okay, cool. Yeah, so I'll talk about this collaboration with a bunch of people. I'll just mention the main ones. Jordi is a person I've been supervising, and he did most of the analysis you'll see today. And then there's several people, mainly computational biologists in the States, who helped us to motivate and also validate the study. So I'll present this package, really. It's called IsoBase, which is a tool to do inference from proteomics data at the isoform level. So the package is already available on GitHub, and we submitted it to Bioconductor, so I think it should be there as well in a few weeks. Let me give you a brief motivation of the study. Intuitively, in proteomics, you want to study proteins, but in bottom-up proteomics, you don't observe proteins directly; you observe peptides. Now, to me, the main two challenges are due to the fact that many or most of the peptides are actually associated to multiple proteins. So when you see a signal at the peptide level, you don't really know which protein or which proteins it's coming from. And secondly, even peptides themselves could actually be erroneously detected. So you're not even that sure about the kind of information that you see. Now, the first issue, just to make an example, a comparison with RNA-seq data, is similar to the multi-mapping issue you have in RNA-seq reads, where a read can be compatible with multiple transcripts. But in proteomics, this is to a larger degree because you have many fewer peptides than you do have RNA-seq reads. Now, as a consequence, what happens is that inference at the isoform level is quite challenging and can often be inaccurate because you have too much noise. What we try to propose here is a method that does inference at the isoform level, but it also enhances the information available so that we don't not only use proteomics data, but also transcriptomics data. Now, just to clarify, transcriptomics data is not strictly required, so you can run the method without it. But we do see, which is quite intuitive, that if you add this piece of information, then inference is more accurate. Now, I spoke about inference, which is a very vague word, getting more into the specifics of what the current package does. We do infer presence absence of individual isoforms and provide a measure of uncertainty with a probability that an isoform is present. We infer the abundance of the protein isoforms, and again we provide a measure of uncertainty like credible interval. And we also try to identify the isoforms where the transcript and protein relative abundances significantly differ. The key to, say, intuitions are that the mRNA abundance is used as an informative prior for the protein abundance, and that the peptides are somehow the information abundance of the peptides is allocated back to the proteins of origin. Now, I say abundance again, which is a big word, because the method actually works both popular measures of abundance in proteomics, which are PSM counts and intensity. So whatever the user provides. Okay, getting a bit more mathematical. This is the input data that we have, and this is the output that we want to obtain. So I'll briefly describe that. As I said, we observe peptides, we want to study proteins. So for each protein, you have a measure of the abundance, again, intensities or PSM counts. And then each protein is associated to, each peptide, is associated to one or multiple proteins. And so you need to have like the equivalence classes in RNA-seq, a list of proteins that each peptide is associated to. And then we also have an estimate of the probability that a peptide is erroneously detected. And optional, but again, highly suggested, you can also provide TPMs, which are used to obtain the relative abundance of the transcripts. Now, starting from peptide and transcriptomics information, we want to observe the overall and relative abundance of the proteins. So how do we do that? Now, assuming that, say, you know that you have a certain amount of abundance, again, PSM count intensities, across all the isoforms in your model, that's like the N here or the summation. Now, this is distributed across all the isoforms according to a multinomial. And this is a very, I think, natural and reasonable assumption. And it's distributed according to this probability vector pi, which indicates the relative abundance of the proteins. Now, if you do provide the mRNA abundance as well, then here is where it enters the model via an informative prior for the relative abundance of the proteins. So it's very intuitive, actually. The relative abundance of the proteins is set to be proportional to the relative abundance of the transcripts. So we don't fix to it, we just push this abundance towards the transcript information. Now, this would be the end of the model if we actually knew the overall abundance of every protein. But as I said before, the actual challenge is that we don't observe protein abundances, we observe peptides. So the challenging part is that we try to transfer peptide abundance to the proteins. And so we use a two-layer latent variable model to recover this protein abundance. So in the first layer here, we sample if a peptide has been correctly or incorrectly identified. And so that is given the probability that a peptide is erroneously identified. So it's quite smooth. Now, only when a peptide was sampled to be correctly identified, then we allocate its abundance to the proteins of origin, where obviously this is again a multinomial, but we don't have all the N proteins that we have in the general model. We only have the subset of the proteins that this particular peptide is compatible with. Now, getting back to less mathematical structures, we validated these on real data directly, and we use a multi-protease approach. So we basically collected the same data set with 6 distinct proteases. I mean, I say we, it's really our collaborators. And then we use something like a cross-validation approach. So we analyzed one of the proteases at a time, and we use the remaining 5 to build something like a ground truth, which is obviously not perfect, it's noisy, but in my opinion, it's actually very accurate. So how do we build this ground truth? We look at the subset of peptides we're really confident about. So we look at those that have a very low error probability and those that map one-to-one with a protein. So where there is no uncertainty in the mapping between the peptide and the protein. And this allows us to build a noisy but accurate ground truth. And then we benchmark our method with and without the mRNA abundance, so in the two forms I described before, and a few competitors. So these four are actually the only ones I found that do this kind of inference. I mean, if there are more that you know of, I mean, happy to hear them. And one of them was actually excluded because of the runtime. So I said we validated. How do we do that? Well, for the presence and absence of isotherms, we make a ROC curve. It's quite smooth. For the abundance, we consider the correlation of the log 10 abundances. Just keep in mind that we don't fit these competitors with the TPMs because there is no way, at least I found no way to incorporate the TPMs in those models. And also, we only compare them in the ROC curve because they don't provide abundance information. So they just don't tell you the abundance of the isotherms. Okay, so here's the first key result. You see at the bottom here, there's a couple of methods, P and FIDO, and then there's Epiphany, and then there is isobase, our approach, without the mRNA abundance. So this is only using the same information as the others. And then, as you add the mRNA abundance, as we were hoping for, you do get an additional increase in performance, which is reasonable because you have more information to work. In terms of abundance, you can see here you have the estimates of our method versus, again, the noisy ground truth, and they have a good correlation of about 72%, which is, if you think about it, it's not too bad because the ground truth itself is noisy. This is not a simulation. Again, this is taken from a subset of the peptides. Computationally, I know I'm running out of time, so I'll be quick. Computationally, the method it runs in a few minutes, okay? 2, 5, 10 minutes, depending on the size of the data set, which I think, again, is quite reasonable, and it's very similar with respect to Epiphany. I'll just say that we have a second multi-protease data set where we're currently analyzing, and results are quite coherent. And again, I'll just remind you the package is on GitHub. I think it will be on Bioconductor soon, and we'll also have a preprint in maybe a couple of months or something like that. Let's see if I can unshare. Okay, here.
Thank you. Is there one question?
Oh, go ahead.
Oh, the ceiling mics will pick it up.
The ceiling, okay,
Yeah.
Oh.
right. Sorry.
I'm curious if this method can be applied to identify any isoform-specific post-translational modification of proteins.
I think you need more information for that, right? I think this is just pointing at, identifying isoforms that are present, abundance, and as I said, also trying to look for the isoforms where there is something that differs between the transcript and the protein abundances. But then that difference could be due to several reasons. As you say, it could be translation, post-translation regulation, or other things. So I think you need more data to understand the reason why you observe a difference. It could give you, as you say, it could give you candidates for that. Let's put it that way.
Thank you. And is Mingbo Lee? Okay, you still in Sydney? You still in Sydney? Oh, sorry. From the University of Melbourne, from WeHi, is going to now present linear models and empirical-based methods for protein-wide label-free quantification differential expression in mass spectrometry-based proteomics experiments. I changed the title. Oh dear, sorry, I still have the older one. How can we get to this? Yours is the PowerPoint, right? Yes. Okay.
Good morning. Good morning, everyone. Thanks for joining the session. As you might have noticed, I've changed the title because it was a mouthful and I wanted to be sound like less technical. Nonetheless, I'm going to talk about our recent work on the missing value problem in proteomics data. So, for a brief introduction, mass spectrometry proteomics is a technology to quantify relative protein expression by analyzing the peptides derived from the proteins. So a typical workflow will start from collecting the samples from which the protein content will be extracted, and then they will be digested by a protease to produce a peptide mixture. And then the mixture of peptides will be separated by liquid chromatography and then analyzed by the mass spectrometer, from which the mass spectral data are generated. And then peptide can be identified and quantified based on the mass spectral data, and protein quantification can be achieved by summarizing these peptides. But when analyzing the label-free proteomics data, one of the biggest challenge arises from the widespread missing values. So very often we observe 10 to 50% overall missingness, with up to 90% peptides or proteins being missing in at least one sample. And missing values are a concern to us because if we just leave the missing values as innate, we lose information from the data. And also imputing the missing values would lead to the loss of error rate control. So either way can lead to biased results and will mislead our downstream biological interpretations. So currently, a lot of the existing methods treat the missing values in label-free proteomics data as either left censored or missing completely at random. So by left censoring, we can model the detection probability by this step function, where if we have the intensity of a peptide is below this threshold, this value must be missing from your data. And if you have a peptide intensity that is above the threshold, this value must be observed. So alternatively, a lot of the methods also assume the missing values are missing completely at random, where it essentially means the probability of detecting any sample, is the same, is a constant for any peptide of the experiment. However, is this what we observe in the real data? And the short answer is no. So here I have a single-cell proteomics data set where on the plot, each point is a peptide. So here I've plotted the average observed intensity versus the proportion of detected samples in each peptide. And from these scatter points, you can already see a positive correlation between intensity and detection proportion. Furthermore, if we zoom in, we can see there are more missing values. So the detection proportion is lower in these low intensity peptides. And so it means the missing values do not just occur at random. And in the same time, we can also see some missing values in these high intensity peptides. So that missing values do not come from censoring either. So to better model this relationship, we've next fitted a logit linear curve for the detection probability, where the logit of detection proportion is equal to beta naught plus beta 1 times the intensity. So from this fitted curve, we can see that detection proportion increases as the average observed intensity increases, which in statistical terms, it means that missing values are missing not at random in label-free proteomics data. So based on these observations, we next propose a probabilistic model for detection, where for each peptide or protein, we assume that the observed values are from a normal distribution with the mean of mu observed and variance of sigma squared observed. And these two quantities are can easily be estimated from the observed data. So with a bit of maths, and we can show that just based on this normality assumption, we can write the marginal log odds of detection as this highlighted equation here. And this defines the detection probability curve, or DPC for short. So the interesting bit of this DPC curve is that it actually allows us to estimate the missing data distribution based on only these observed quantities, mu observed and sigma squared observed, as the DPC actually estimates the probability of observing a sample given its underlying intensity, even when this underlying intensity is not observed due to missing values. So next, we've fitted the detection probability curve on the example data set. So you can see a dashed curve. This is our starting curve, and we've obtained that from only observed data. And then with the DPC, final DPC curve, which is the blue line, we corrected the starting curve by estimating the real average expression in each peptide after accounting for the missing values. So one of the important interpretations of this DPC curve is that the slope of this curve, or beta 1, actually signifies the amount of statistical information contained in the missing values. As beta 1 goes bigger, the bigger beta 1 is, the more informative the missing values actually are. And in addition, what's also cool about the DPC curve is that it, in fact, summarizes the two popular cases I've just mentioned. So when the slope is 0, or beta 1 is 0, we have the detection probability equal to beta naught, which is a constant. And in that case, we have the missing completely at random. And also, as beta 1, or the slope, gets bigger, the curve will get steeper. And eventually, this DPC curve will become a step function when the slope goes to infinity. And in that case, we have the left-censored missing values. So we've provided the DPC as a probabilistic model for the missing data. And next, we would want to use the DPC to inform the differential expression analysis. And we've actually experimented with a lot of potential options. So for example, if we apply the DPC on the protein level, we can perform principal imputation. However, our experience show that it is a useful approach for data exploration, but probably not the best for DE analysis. And alternatively, you can also formulate the DPC into a likelihood ratio test, and without the need of imputation. And another option is to apply the DPC on the peptide level. So in particular, if we consider the protein quantification problem, where we need to summarize the peptides into proteins to obtain the protein expression levels. So we're using the DPC to represent the missing values and fit an additive linear model by maximizing the posterior with a diffuse prior. And after getting the protein expression levels, we can then perform a customized limit analysis for differential expression. So in summary, so we've shown that missing values are missing not at random in level 3 proteomics data. And to account for that, we've proposed the detection probability curve to model the non-ignorable missing values in the data. And we also point out that DPC needs to be incorporated into the differential expression analysis to avoid biased results. So the DPC bit of the analysis is already available on GitHub through this repository. And we provide a very simple quick start guide to help you investigate the missing data patterns in your own proteomics data sets. Meanwhile, we also aim to release the complete pipeline on Bioconductor as soon as we can. And with that, I'd like to thank my wonderful supervisors, Gordon and Andrew, and as well as our funding sources. Thank you. I'll be happy to take any questions.
One quick question. I think you said, if I caught this correctly, that imputing the values would lead to a loss of error control. Could you comment on that? Why is that?
So in theory, single imputation methods are problematic.
Sure, but if you did multiple imputation, right?
That is an option, yes.
Okay.
But in practice, it will be kind of tricky to implement. And we will also have to take into account the cost of computation.
Okay.
Yeah. Thank you.
That looks like a great resource. And finally in this session, I invite Samuel David and Boa Tooze to talk about BugFizz, sorry. A harmonized data resource and software for enrichment analysis of microbial phylogenies. And you're in New York with the Waldron Group.
Yeah, exactly. Thank you. It's 9 minutes, right? Hmm? It's 9 minutes. So, yeah.
It's PDF, is it? This one?
Yeah.
We need to make this big. How do we make this big?
I think it's 11 maybe. Oh. All right. Thank you. Well, hi everyone. Happy today to speak today about bugfish. Yeah, before starting, I would like to give credit to Kelly, who's among the co-authors, because she's the co-lead of this project. So thank you to her. Yeah. So I would like to start speaking about a little bit about set enrichment analysis, which is a computational method commonly used in transcriptomics. Which is mainly used to identify overrepresentation of pathways of functions in a list of genes. So, for example, you can have two lists of genes, but you can't kind of summarize which processes are overrepresented in them. And this makes the interpretation of this list easier, and they also make it easier to compare to other studies. So for that, we need we need a molecular profile of that data. We also need a database of gene sets. And in the case of genes, I think their reference is MSigDB, is one of the most popular. And you can also create your own sets. You have a good annotation, for example, with the gene ontology. I think the biological processes to go to. And yeah, and you can apply these methods also to microbiome data. For example, with the bug set enrichment analysis. And one of these databases that is kind of an analog to MSigDB is BugSigDB. I will talk a little bit more about it later. Yeah. So as I say, you can create your own sets. You have good annotations. And currently, we were interested to know in physiological data about microbes, which is currently dispersed among different resources over public databases and sources. And this is an example in this heat map. You can see some true references here for different types of physiologies. You can see that every source provides different types of information at different taxonomic ranks, and they're missing some. So we would like to fill in these gaps. And we also would like to harmonize the different terms they use. So that's how we started working with Bugfish, which provides harmonized annotations, which can be turned into signatures for microbial physiologies. And we have a controlled vocabulary for these terms based on ontologies. I'm not citing anything here because we have different sources of ontology. And we would like to make these data findable, accessible, interpretable, and reusable fair. So on the right, you have some kind of loosely the workflow that we're following. We download the data and harmonize it. We resolve any conflicts that could be in the annotations between different sources. And we map all of these annotations to a tree data structure that we have based on the NCBI, and we propagate this. And we convert this back to a single tidy data frame, and we export this file. Currently, it's only living on GitHub as a developed version, but we're going to have a release on Synodo soon. You can find the data there as CSV files or GMT text files. This is an example of how the data looks like. I would just like to highlight like 3 columns here to understand the rest of the figures, which are the NCBI IDs, which are basically the tax IDs of the NCBI. We have the attributes, which are the actual values that will define the sets or the annotations. And these are kind of grouped into attribute groups. And yeah, and we, these are kind of similar. We also use these attribute groups to enclose mutually exclusive attributes. So, yeah. So we performed the propagation of our initial set of harmonized terms. We went from 50,000 to over 1 million. We covered from 16,000 to over 74 taxa. So it was, well, we got a lot of annotations through taxonomic propagation. You can see on the right this heat map. This on the rows, you can see like the 32 physiologies or attribute groups, and on the columns, different taxonomic ranks. So we went from having very little and strain species and genus to having a lot for some of the physiology. So the performance wasn't the same for everyone, but I think it's relatively good. Yes, we performed some AOC curves with a holdout to present the annotations. And the curves, at least for one of these, I think they look good. We tried different holdouts with different taxonomic grants, and yeah. And yeah, we also provide an R package to access this data. It's going to be submitted to for the next release of IOC. It's basically two functions that matter the most. One is for import bug fees, in which get this tidy data frame. And the other one is the get bug fix signatures, which give us the signatures, which can be easily integrated in different workflows already existing in R. You can see on the right an example of one of these signatures. In this case, optimal pH. Yes, and we used these signatures for a couple of analyses or use cases. One is with a ground truth data set coming from the Human Microbiome Project, comparing subgingival versus supragingival plague. And the ground truth of this was the enrichment of aerobic taxa in the supragingival plague and the enrichment of anaerobic taxa in the subgingival plague. So we used GCA using a package enrichment browser, and we recovered the expected result, as you can see in the scores here. Positive scores are for aerobic and negative for anaerobic. So, and we also use this with the BoxDB database that I mentioned at the beginning. You probably know it's kind of new, so probably not fully familiar with it, but it's a collection of publicly viable signatures, and they're manually created, and they come from over 600 published studies. So most of them, I think, come from sets of differentially abundant taxa, and they span different body sites and different conditions or diseases. So we use all of our sets and bug fix and compare all of them against all the sets. BoxDB using Fisher exact tests, which is a kind of overrepresentation. And yes, we got like 153 BoxDB signatures that were significantly enriched with this FDR threshold, and about 144 bug fix signatures. And yes, just briefly showing a heat map here, how presenting the body sites and the different types of conditions or diseases that were enriched with some signatures in bug fix, right? But there are a lot. We're still going through all of them. But yeah, one example was that we got, for example, bug fix signatures related to disease enriched in tools of samples of patients with COVID, which is what we would expect. Some of the signatures were related to infection, pathogenicity rating, biosafety level, and pathogenicity in humans. So in summary, BoxFix provides harmonized microbial physiology annotations. We provide an increased completeness of these annotations with different taxonomic ranks. And we expect this will allow an easy integration with enrichment workflows already existing in R and outside of R with the GMT files. And this, I will try to abide to the FAIR principles for data sharing. Yeah, and that's it. Thank you, everyone. And some links there, in case you are interested. Yeah.
Any questions? I have just one, if I might. So for the different attributes, is everything mapped into the ontology, so it's hierarchical?
Yeah, it's not hierarchical. We kind of made them to be on the same level. So yeah, and we didn't have an ontology for everyone. So the ones that we could map to ontologies, they have ontology, but some others. We harmonize the vocabulary, but they're not mapped to ontologies. Yeah. Thank you.
So let me just stop sharing this so we can see each other. And so with that, I would bring up so everybody can see each other. No, that's not working. Right. Okay, so everybody can see each other. And with that, I'd like to bring this session to a close, and I really want to thank all of the amazing speakers. Could you just go on stage, Leo?
I think, yeah, there's a question.
Oh, sorry, Leo.
Okay, thank you. Well, about this last talk, I was just curious to know if it's easy or if it's possible in this system to create your own sets, custom sets, and integrate somehow into the analysis workflow. Thank you.
Yeah, well, the workflow that we are using with the enrichment browser, this is a specific case of differential expression and enrichment analysis. So you have your custom sets based on differential expression, but yeah, we gave some options for filtering, so you can map these annotations to tax IDs. You can create your own sets. You have a background also to compare against it, which is based on NCBI tax IDs. You can create. You should be able to create your own sets for analysis. Thanks.
Could I just add that Samuel has made a package called TaxP Pro for the propagation components of this, which are the main thing that probably you would want to add to your own sets, because the sets themselves, they're just lists. But Samuel's trying to make it easy for you to do these other steps as well.
That's great to know. I've been posting links to all the packages in the Hopin platform. So for people who want to get the links later on, they're there. Levi, if you could post the link to that in the Hopin platform, it might be a useful resource for people who want to have a look at that.
Yes, let me just find it.
Oh, that's brilliant. That's really, really good. And I think it's a great resource. Analyzing, these complex data sets. These kind of resources really, really facilitate that. I apologize again for the late start to this session. Sorry, that was my fault. There was a miscommunication there, so I'm really, really sorry. And with that, I would like to bring this session to a close. And I think this has been a really interesting session with amazing talks, spanning proteomics and microbiome research. I think the importance of this area is really highlighted by the geographical diversity of the talks. We've had talks from Italy, Finland, Australia, and the US here in New York. And I would like to thank all the speakers again. Thank you.